Hi everyone, welcome to the third seminar of the One Health Seminar Series. This series is organized by the One Health Institute at the University of Guelph, along with graduate students enrolled in the One Health Collaborative Specialization. So my name is Sydney, and I'm an MSc student in the Department of Population Medicine, and I'm also one of the students who's enrolled in One Health. It is my pleasure to be facilitating this third seminar of the 2021 to 2022 academic year. With great respect, I would like to take a moment to acknowledge that the University of Guelph and many of our homes resides on the ancestral lands of the Adirondack people and the treaty lands and territory of the Mississaugas of the Credit. We uphold the significance of the Dish with One Spoon covenant and the continuing relationship with our Indigenous neighbors. We recognize that this gathering place and all the places that we are joining from today is home to many First Nations, Métis, and Inuit peoples, and by acknowledging them, this reminds us of our relationships to this land where we explore, share, and discuss and learn about our shared responsibility for human, animal, plant, and ecosystem health. It is also important to remember the role that Indigenous peoples and their knowledge play in our understanding and approaches to One Health. For any of those wishing to learn more about the history of Indigenous peoples of Turtle Island, the University of Alberta also offers a free 12-week online course that delves into Indigenous histories and contemporary issues in Canada. So before I introduce our speaker today, I'm just going to take a moment to inform everyone that this seminar is recorded, and this recording is posted to the One Health YouTube channel, and a link will be shared on the Institute page. So this recording may also be used for promoting and showcasing One Health work being done on campus. If you have any questions or concerns about the recording of the seminar or just One Health in general, you can contact One Health at uoguelph.ca, and I'll also post this in the chat momentarily. At the end of the seminar, there is time for questions and discussion, so you're welcome to ask your question by using the raise hand function and being invited by the facilitator to speak, or you can post your questions in the chat and then the facilitator will read them. Now it is my pleasure to introduce our speaker today. Dr. Brian Husband is a professor in the Integrative Biology Department at the University of Guelph, and he runs the Plant Population Evolution Research Lab. Today, Dr. Husband will be introducing us to the topic of feral apples, and we'll be discussing whether this problem can be viewed through a One Health lens. So without further ado, I'm just going to turn this over to Dr. Husband.
Thanks very much, Sydney. I'm going to share my screen right away here. Everything visible there? Okay. Well, thanks so much for that introduction, and thank you for the opportunity to participate in this series. I have to say it's not something that I had planned on, in that I, like many faculty on this campus, maybe don't necessarily think of my research program as falling neat and tidily within the One Health framework. But the more I thought about it, the more I thought, oh, I think I have maybe an argument here. And so I'm looking forward to sharing a little bit about my research program, or at least one component of it, and getting your feedback about how you think it fits within that One Health framework, and even to think critically about what kinds of dimensions could it have in order to strengthen the One Health approach that we could take to this problem. So what I wanted to do is just kind of give you a bit of an overview of the research that we're doing related to apples, as you mentioned, Sydney, and its relation to unmanaged ecosystems and native species that are in the vicinity. And so what we're really talking about here is sort of ecological sustainability of agricultural or food systems, and that's kind of the context I'm going to put my presentation in today. So the title says, "Is this One Health?" because I'd love your feedback in terms of how well you think this fits, and I'm happy to discuss the approach that we take, as well as some of the key results that we found. So you also mentioned the name of my laboratory. This is actually not widely known, so I know somebody's done their homework. The lab, I define it as Plant Population Evolutionary Research Lab, and its primary focus is on the ecological and the genetic or genomic aspects of plant populations. So I suspect you probably haven't heard too much about plant populations within your One Health series yet. So this will be something completely different for you. So yeah, many of the students in the lab are combining the ecological and genetic perspectives to understand plant populations. So in a sense, it's sort of multidisciplinary at that stage already. We have a focus on plant reproductive biology. And what I mean by that is everything from sort of gamete production, gamete quality, fertilization processes. I'm really interested in mating systems and who mates with whom within plant populations, and ultimately how genes are transmitted from one generation to the next. So the reproductive system is a critical kind of controller in a plant species machinery, the biological machinery in controlling the way genes move on a landscape and the way they move from one generation to the next. And so that's why we really do focus on the reproductive aspects. And ultimately, we use the tools from ecology, genetics, and reproductive biology to address a number of different problems or questions in biology. Some of them are, and I would say a great deal of what we do is designed to understand fundamental processes in evolution, whether it's species formation or adaptation. So we spend a lot of time working on those sort of fundamental problems. But we also have an opportunity to address some more applied issues in both agriculture and in conservation biology. So we have different research threads that are addressing both of those things. And it's the agricultural context that I want to talk about today. Whoops. So I think this one slide is trying to capture kind of the perspective that we take on food production and agricultural systems in general. We're interested in that. When we talk about sustainability, we're interested in this case in the ecological sustainability. That is the impact that agricultural systems and food systems have on surrounding processes and biodiversity. So specifically, what we think about is the impacts that agricultural systems have on unmanaged ecosystems. And there's all sorts of ways in which our agricultural systems can influence that. I've made a brief list on the left-hand side here. For one, land use patterns and the development of agricultural systems on the landscape obviously has a big impact on the distribution of unmanaged or natural habitat. It creates a very fragmented landscape, as you can see in that top picture, where we have lots of edges where agricultural systems come into are adjacent to less managed or unmanaged systems. So there's lots of opportunity. There are lots of edges, if you like, for interaction between those two ecosystems. The activities in agricultural systems, of course, can have huge influences on the abiotic environment, both, for example, nutrients, moisture levels, etc., as those agricultural systems interact with the adjacent unmanaged systems. And one of the areas that we're particularly interested in is the direct impacts on native biological diversity. So what I'm talking about here is the impacts of domesticated species, crops, on native biological diversity. And that can happen a couple different ways. One, when crops can become naturalized so that they become established outside of cultivation. We talk about feral populations of domesticated species that can interact directly with other species in the unmanaged ecosystems. And also, even if they don't establish outside of cultivation, they can still interact with those species through things like hybridization. And this is, of course, important from a regulatory point of view, because it does potentially mean that crop species can be very significantly influencing native species in the near proximity to them. So if I'm going to take my research and place it within a One Health kind of framework, this is what it might look like. I'll start just with the three kind of core pillars that we talk about within One Health: human health, ecosystem health, and then some key species, animal or plant, or I suppose even microbe health could be on that bottom right corner. And collectively, the interactions between these three pillars can provide us with insights about health and about managing health that we might not have by examining any one of these pillars individually. So in our research, the One Health challenge that we're really trying to address is all about how to manage agriculture in an ecologically sustainable way. That has many dimensions to it. In our particular case, the primary focus around human health is related to food quality, food production, food security, so that everything related to the management of food systems. But at the same time, we recognize that the decisions that we make with respect to food system management can have potential impacts on the health of native biodiversity, on unmanaged ecosystems, and on specific species, or for example, species at risk within those unmanaged ecosystems. So there's this very complex network of interactions that we often don't think about when we're so focused on managing our agricultural systems. In my own specific research, basically the food system that we're focusing on is apples. And I'll talk to you about a number of different dimensions of that work. But what I can say is that as we became involved in research on apple production, efficiency in an agricultural environment, we soon realized that we would be remiss if we didn't start to think about what the impacts of those systems are on the neighboring ecosystems. And that's when we started to get interested in native biodiversity, and specifically, I would say a couple of different species that were of greatest interest to us. One is not just one species, but pollinators in general, wild pollinators in particular. And I'm not actually going to talk too much about the wild pollinators today, but that they play an important role in this and are influenced by the presence of the agricultural systems. And then the second is close relatives to the domesticated apple, other apple species that are in our forest environments that have the potential to be affected by the presence of the domesticated apple. And of course, it's more complicated than that, because we can talk about those three different pillars and those interacting components, but of course, the stakeholders, the beneficiaries of thinking about health of our food systems and their interactions with native ecosystems involve, a variety of different people and organizations that are affected by those decisions. And so I'm just kind of showing you this because it means when we're doing this work, we need to be working closely with the experts, if you like, and the people that are going to be affected by the decisions that are made in any one of these three pillars. So you can see there for the food systems themselves, the apple growers that we deal with most directly. They're the regulatory agencies like the Canadian Food Inspection Agency, crop breeders who are interested in processes for developing better apples, but also the availability of germplasm, a genetic material that's useful for breeding that might be found outside of the crop itself. And of course, others out there that are both landowners that might have apples on their property or simply consuming this particular food. So there are other agencies and organizations that would be affected by whatever decisions are made with respect to food systems, but that also have interests both in ecosystem health, such as conservation organizations, and the status of particular species. So what I want to do is just kind of take you on a little bit of a tour, somewhat of a loose chronological tour of kind of how our work unfolded. And I'll present to you a little bit of, talk a bit about our approach, and I'll provide some highlight results just to give you a bit of sense of what we've been uncovering as we go along. So the work that we started with was simply on apple production systems. That's where we started in the early 2000s. We got involved with a number of apple growers, with the Ontario Ministry of Agriculture, Food and Rural Affairs, to try to provide evidence-based approaches to improving the production and quality of apples in Ontario orchards. And that brings me to the key species here, Malus domestica, which is the domestic apple. This is, of course, been around in Ontario for quite a while, but its origins are really in Central Asia. It was introduced around the 17th century Globally, this is a $78 billion industry, so it's a massive, probably the single biggest temperate grown crop in the world. In Ontario, it's about a $600 million industry. There are thousands of cultivars. You hear anything from like 5,000 to 10,000 different recognized varieties of apples that are grown globally. In Ontario, we're talking around 200, and 15 of those are particularly dominant. So that's interesting in itself, in that we see, especially in contemporary apple production, a narrowing of the diversity of genotypes that are used in our apple production. But in Ontario, it's also narrowed for a bunch of different reasons. The apple industry certainly is a challenged industry in Ontario for a number of different reasons. One is climate change, and the apples are growing kind of on the margins of their tolerance in central Ontario, at least. So there are some climate change issues that need to be considered. Pests and pathogens have a huge impact on productivity in apple orchards, and pollination is the other one, because we're seeing an increasing reliance on domesticated bees, honeybees, if you like, and less ability to rely on wild pollinators to do this work. So there's lots of interesting challenges facing this agricultural system, and our focus has been on the pollination side of things. So the primary goal when we started working with apple growers was to find ways to improve fruit production, to bring a kind of evidence-based approach to our understanding of pollination in apple orchards, and to find ways to improve pollination efficiency and effectiveness in orchards. And for the rest of this talk, if there's nothing else that you get from it, this is the key. This is, everything rests on this little piece of information here. And basically, the way that apple orchards are typically operated has two important features. First is that apples are, for the most part, self-infertile. That means that they cannot pollinate themselves. They cannot pollinate themselves, even they cannot mate between trees of the same cultivar. So, for example, here, my little diagram, a Macintosh tree produces flowers. It will not produce fruit unless it gets its pollen from a different variety. So it doesn't matter if there's an orchard that's 2/3 of it full of Macintosh trees, they cannot produce fruit unless you introduce another genetically distinct cultivar that it is compatible with, and then it will produce fruit. So that's key. And then that is kind of coupled with the real challenge, and that is that the way apples are typically grown, the cultivars are grafted or cloned, if you like, so we can produce multiple copies of the same cultivar. And we tend to grow them in large blocks. And that's because each cultivar has a different management need, different pesticide treatments, different pruning treatments, different times of maturity, etc. So growers typically grow them in these blocks, and you can see the conflict there, that if you grow them in big blocks, you're basically creating a pollination environment that is ineffectual for producing fruit. So we got involved in this, and in essence, what we've been doing is using genetic tools and paternity analyses to track how pollen moves in orchards and to better understand how that happens. So yeah, we can take the seeds from the fruits that are produced in the fall. We can do genetic analyses on them and infer from that who the fathers were. So which cultivars are siring the seeds in which other trees. And we can learn things like is shown in this diagram. It shows how the cumulative proportion of seeds from a given position in the orchard, how that the proportion of seeds that are sired by trees in that particular position changes with distance from that tree. So you can see that with about one row over, about 35% of the seeds that this particular cultivar sires in the orchard are in that next row over. a smaller percentage of sort of diminishing returns as you go farther away from that tree. So the pollen's only getting a certain distance in the orchard. And the take-home message from that is that most of the pollen that a particular tree receives in the orchard is about 20 meters away. So we really need to think about what the composition, that is the cultivar composition, is within the orchard, thinking in terms of that spatial constraint, because that's where most of the pollen's coming from for a given tree. This also shows us that the distance that pollen moves in an orchard is about the same, whether it's up and down the rows. You can see in this orchard a clear row, and there's long been this belief that pollinators move up and down the row way more easily than they do across. And that's simply not true. The pollinators are moving across rows just as easily as they are within rows. And then once we knew that sort of the spatial dimensions of pollen dispersal, we could start to look at things like cultivar diversity and cultivar compatibility, because not all apple cultivars interact with each other to the same degree. And we can start to ask, how important is cultivar diversity in promoting food production? So if you think about this tree here and the 20-meter radius that I just talked about, we can think about what's the diversity of cultivars within that, and how important is that? And what we see from these two figures on the left-hand side is that diversity of cultivars matters a lot. That if you're in an orchard with low species diversity, a given amount of pollinator activity, so if this is what's on the x-axis here, the more pollinator visits that the tree receives, the higher the fruit set is. But that relationship is much shallower when there's low cultivar diversity than when there's high. So we need about half, if not a third, of the bee visits in a diverse orchard to achieve the same level of fruit set. So these are the kinds of things that we were really focused on in trying to understand how to improve apple production within an orchard. And so a lot of these kinds of findings have influenced sort of guidelines for growers in terms of the combinations of cultivars that they grow within orchards, the spatial arrangement of those cultivars, and also the placement of bee colonies within orchards to get the maximum pollination benefits for their buck. Okay. So we've been working on that for several years, and we started to think, okay, we can think about pollination within apples, but what's happening outside of the apple orchard? So this is now where we're starting to branch out and think about what's happening in the surrounding areas. What's happening outside of the apple orchard, and can the apples themselves influence native ecosystems that are adjacent to them? This becomes very important from a regulatory point of view, because it does speak directly to the ecological impacts that this crop has, whether it's potentially impacting neighboring species in a negative way. And I produced this image of the Arctic golden apple. Some of you may be familiar with this. This is actually one of the few genetically engineered apple cultivars that's kind of been developed in North America, and it has received approval in the US. It's received approval for sale in Canada, but is not currently being grown on a large commercial scale in orchards. This is a non-browning apple, so it's been genetically modified so that it doesn't brown when it's exposed to air. And there's been a great deal of controversy over whether these kinds of modifications should be grown on a large commercial scale. And one of the arguments is that these kinds of cultivars can have negative impacts on surrounding species, and potentially the genes associated with these apples could actually escape and become incorporated into neighboring close relatives of the domestic apple. So enter the next player in this. It turns out there is a native malus, a native apple in Ontario, and it's Malus coronaria. We call it sweet crabapple. There are two different native Malus species in Canada, but there's only one in Ontario. It has, you can see it here, the distribution's relatively restricted, although it comes up into southern Ontario and then down into a handful of northern states. It has flowers that are very apple-like. The tree is actually more of like a shrub. It's a short tree, shrubby looking thing. You can see that in the middle image. And it grows in these kind of open forests that you see in the lower image there. Often shallow, we call them alvar habitats, that have shallow soils where the forest is a little bit more open. There's all sorts of interesting opportunities for interactions between the domestic and the native crabapple. And you can see this map here where the hatched bars show you the distribution of commercial apple production, and the gray zones, the distribution of the native apple. And so there's lots of overlap there. And it's even more extreme than that, in that turns out that domestic apple has become naturalized. So there are feral populations of domestic apple in southern Ontario, and in fact in central Ontario, and even further north than that. I wouldn't say northern Ontario, but it's certainly on the southern margins of northern Ontario. So domestic apples have become very widespread, and these are just a variety of images of these apples. You'll see them in open fields, you'll see them on rail lines, you'll see them in a wide variety of kind of disturbed or open habitats, which turn out to be quite similar in habitat to where the native apple is found. So the first question was, is there any evidence that domestic apple is interacting with these native apples through pollen dispersal and hybridization? So here's an open habitat you can see, and there's a little crabapple growing on the left-hand side and the giant big feral domestic apple. So these are not cultivated apples. They've escaped cultivation, and they're now a self-sustaining population within our unmanaged forests. So we wanted to know, is there any interaction between them? We know, and I'm not going to show you this, but they share a great deal of pollinators. The pollinators move back and forth between these on a regular basis. So here's the most complicated data slide I have for you, but you can kind of ignore all of it except the left-hand side. But I would just say is what we've done is we've used some variety of genetic and genomic analyses to examine the seeds produced by the native crabapple. And again, using a kind of paternity analysis, we can figure out who is siring those seeds. The whole story becomes a little bit more complicated. And this figure on the left kind of shows this, that when you look at the genome sizes, or what we call DNA ploidy, so the number of copies of each chromosome set per nucleus, you can see that there's a highly diverse set of seeds that are being produced by the native crabapple. And there's a long story here that the particular ploidy level, from diploid to triploid, tetraploid, pentaploid, hexaploid, heptaploid, and octoploid seeds. So these are producing this incredibly diverse range of types of seeds. And it turns out that those ploidy levels are determined by the reproductive origin of those seeds. So that is whether the seeds are pollinated by domestic apple versus crabapple, whether the seeds are asexual versus sexual, and I can happily talk about this, but I'm not going to give you the gory details here. But some of these seeds are entirely asexually produced. And it also depends on whether the gametes of either the mother or the father are fully reduced, as you'd expect with meiosis, or if they're unreduced. So there's some errors in meiosis and the gametes are not going through double reduction. So that's a complication you don't need to know. But what you do need to know is that these two groups here, which are the triploids and the pentaploids, can only be explained through hybridization with domestic apple. And it turns out that a pretty significant number, almost 30% of the seeds produced by this native species, are sired by domestic apple. So this really threw us, we were used to seeing numbers that are 1/100th of this for other species. And of course, this was of great interest to the Canadian Food Inspection Agency because it does suggest there's potential for gene flow or gene escape from domestic apples into native. And this, of course, led us to ask a series of other questions, which was, well, if there is hybridization happening, what is the impact? The hybridization itself isn't necessarily bad because these hybrids may not go anywhere. So what are the impacts? And so we've gone through a whole series of studies over the years looking at the genetic impacts, the demographic impacts, and the reproductive impacts of hybridization on native crabapples. And we've done this through a whole bunch of different studies involving experimental pollinations, DNA fingerprinting approaches, and what we call flow cytometric seed screening, which is just another way of looking at the ploidy level of different tissues in the seed. And from that, we can infer what their reproductive origins are. So I'm just really going to quickly give you sort of the highlights of what have come from this. The bottom line is that there is no evidence of genetic assimilation. So when we do the genetic analyses, we can create a plot like this. This was called a structure plot. And what we've done here is include a whole bunch of the native crabapple and then a bunch of reference cultivars. So all the cultivars we can get our hands on that have been grown in Ontario, we genetically characterize them. And then this analysis basically tells us whether there's any genetic divisions or clustering within that data set. In this case, those genetic clusters are represented by colors, and there are two dominant colors, the yellow and the orange. And as you can see, the two colors are exactly coincident with the two different species. So Malus coronaria has a completely different color than all the reference cultivars, signaling that they're genetically different from one another. And each one of these vertical lines represents a separate individual, and the colorations tell you whether that individual has any genome that's part of the different genetic subdivisions. So you can see here there's no evidence, or virtually no evidence, of orange showing up in any of the Malus coronaria individuals, and similarly no yellow showing up in the domestic apple cultivars. So we can conclude from that that there's no mixture happening, that even though hybridization's happening, the genes from domestic apple are not being incorporated into the native crabapple. We did a whole other series of studies that basically came to another conclusion, and that is that as the number of seeds that are sired by domestic apple goes up, there is no concomitant decrease in the number of native crabapple that are being produced. So the two seem to be somewhat independent of one another. We're seeing a rise in the number of hybrids, but we're not seeing a corresponding decrease in the number of native apples. So this is good news. It's saying that the presence of domestic apples not reducing the number of native crabapple offspring that are being recruited into these populations. And then lastly, the biggest result in a way is that by looking at the origins of the seeds that are produced when they're sired by domestic apple versus sired by native crabapple, we're seeing a shift in the balance between asexual and sexually produced seeds. And with domestic apple, we're seeing a much higher percentage, actually 100% of the seeds that are produced are derived now through asexual means. So the presence of the domestic pollen is not, it's not leaving its genes there as remnants, but it's inducing a change in the gamete development and causing seeds to become asexually formed rather than sexually formed. And I'm happy to talk about that little detail as well. This is actually quite a significant result that we're still trying to get our heads around, but it kind of suggests that without creating hybrid trees, it's actually fundamentally changing the balance between asexual and sexual. And that's important because when we look at a population of crabapples like this one, you can see these are all individual trees in this property. It turns out that every one of these trees are genetically identical. And so it appears that asexual reproduction has been way more important in establishing these native crabapples than the sexually produced seed. And so the fact that the domestic apples are promoting the production of asexual seeds suggests the possibility that the presence of domestic apple is completely changing the genetic structure of these populations from one of very diverse populations to one of very genetically homogeneous populations. Yeah, I see the time, so I'm going to keep moving fairly quickly. The next step was to explore what the source of the domestic pollen is. So we knew that now we have evidence that there's hybridization going on, that domestic apple is siring seeds in native crabapple, but where is that coming from? Is the domestic pollen coming from cultivars in commercial production, or are they coming from feral apples? The bottom line is it's all coming from feral apples. So the invasion of our unmanaged environments by feral domestic species is putting these trees in close proximity with their close relatives, the crabapple, and they are the ones that are now hybridizing aggressively with those native crabapples. So I'm going to just say that's all I'm going to say there, is that we've used paternity analysis to look at this as well. And this aerial photograph basically shows you an example. Here's a crabapple down here that I've just made pink. Yeah, we took one seed or two seeds from that crabapple and we completed a genetic analysis. And from that, we could infer where the most likely father was for that particular seed. And here it turns out that one of the seeds was fathered by a feral domestic apple that was right beside it. And then another seed was sired by this feral apple that's way over here. So we see the feral apples serving as fathers, and they can be anywhere from 5 to 350 meters away from a given native crabapple. So now, as time progresses, we're becoming increasingly fascinated by the feral apples and trying to understand how they've come about. And the two arguments for most fertilization processes, and there are a lot of crops that go through this process, is this one, which is called the exoferal pathway, which basically says that feral apples or feral crops happen when there's hybridization between the crop and a wild relative. The alternative is that there is no involvement from a wild relative, that these plants are predisposed to growing outside of cultivation, and so that naturalization process is happening without hybridization. And there's a whole series of genetic analyses we've done of this, and suffice it to say that we can find no evidence that the feral apples are hybridizing with the the feral apples are not the product of hybridization between native and domestic apples. And so you can see this is the same figure I showed you before, only now we've added all these feral trees. About 600 feral trees have been added to it. And you can see there's no evidence of yellow, which is the genetic cluster that the native crabapple belongs to. There's none of that that's showing up in the feral trees. So these feral trees are establishing kind of on their own. They are the product of matings between domestic apple cultivars, but not involving native crabapples at all. And this is just another way of presenting this, but when we take the feral apples, we can actually find two very different clusters within them, shown by this diagram with a color coding. And each cluster, the feral apples are associated genetically with a different set of cultivars. So these are likely contributors to the feral apples. So this group, the red group, we have cultivars listed here that are closely related to this set of feral trees shown in the red. You can see a number of them are from US, and others have originated from Russia that have been introduced into Canada because of their cold hardiness. And then the other group of ferals is associated with a different set of cultivars. Some of these are more recognizable because they're still commercially widely grown in Ontario. We've even done parentage analysis on some of these feral trees where it's possible to ask which cultivars are the parents of these particular feral trees. And what we find is that of the 200 and some cultivars that have been grown in Ontario, most of the feral trees are the product of matings between a small number of cultivars. So 99 of the cultivars that we're aware of account for about 70% of all of the parents of our feral trees. And it turns out all of these are heritage cultivars. So these are cultivars that were grown prior to 1905. So this is kind of an interesting historical element because it's showing the legacy of these old cultivars that are not grown very often anymore. And the genomes are residing within these feral apples that are widespread on the landscape. And you can see these here are the 9 cultivars. Some of them are still very familiar: Macintosh, Red Delicious, Northern Spy, widely grown still, but others not widely grown. In fact, we have a hard time finding examples of these trees now, but their genomes still reside within the feral trees. And lastly, I guess I just wanted to say this has led us to a new adventure in this whole research program. By going through this whole process, we've established now a fairly large genetic library of apple cultivars that allow us to now engage with the public and with the apple industry in a way that we couldn't before. So we've created a page on the website called the Ontario Heritage and Feral Apple Project. And we've now engaged citizens, both growers, landowners of old abandoned orchards, and also professional growers, commercial operators. And we can use our genetic tools to do a couple things. First is to help them confirm the identity of their cultivars, because a lot of growers don't know what they have in their orchards. And a lot of landowners have discovered they have ancient orchards on their properties and they have not a clue what they are. So we can go in and do these analyses and help paint a picture of what's in their orchards and what this kind of history of the growth of and the use of apple was across the landscape. And it's turning out to be a lot of fun. And as part of this, we actually are encouraging people to identify the oldest trees, the older orchards, perhaps again abandoned ones, because it also not only helps them, but it helps us expand our genetic database as we begin to look for some of these lost heritage varieties that are no longer grown on a widespread basis in Ontario. So lots of opportunities, but it's actually turning out to be quite interesting, and we're engaging a number of different communities throughout Ontario. We're now working with some growers and organizations in the US and even on the West Coast who are all interested in this kind of a resource. I think just to sum up here, I'll say that, what we've discovered is that by going beyond just our focus on the apple production systems themselves, we're learning all sorts of things about the potential broader impacts of the food system, specifically on unmanaged ecosystems. Through hybridization, we're seeing the potential for domestic apple to drive the shift in native crab apples from the typical sexual produced offspring to asexual, which is having big impacts on the genetic structure of these populations. And of course, feral apples are infiltrating many of these sites and could be potential competitors for native crab apples. So something that we have to explore further. And this information also then feeds back on the way we think about commercial apple production. Our knowledge of hybridization with native crab apples is going to have a big impact on the regulation of newly produced or novel apple cultivars, or how they're managed. So things like the distance that they're grown from native habitat, etc. Also, I think it's changing the way we think about feral apples. On the one hand, they look like they are kind of serving as a conduit for genes from the commercial apples into native crab apples. But there may be benefits to those feral crops as well, in that they might serve as refuge for wild pollinators that can support food production in commercial orchards. And maybe more concerning, they may also serve as refuges for apple pathogens and pests. So as hard as the growers try to control these things, the presence of the feral apples may serve as a sort of an infinite source of these kinds of challenges that they have to continue to deal with. So there you have it. That's kind of my perspective on kind of a One Health view of what we're working on. And I do think that it's making an argument that a focus on simply human health and food systems in particular is not enough, and that when we expand our perspectives and think about interactions beyond those boundaries, we learn a ton that's going to have an influence on the way we manage our food systems in the first place. Ton of people to thank, and including growers, field assistants, researchers. I have to a special shout out to Paul Crone, who's my research associate, who's just done a ton of work in driving this work. And then a whole bunch of people and organizations that provide resources for us.